Deep in a storage drawer at a European museum, catalogued decades ago as just another archaeological curiosity, sat a skull that nobody had bothered to look at twice. It was elongated, unusually shaped, the kind of specimen that gets a shrug and a shelf number. Then someone finally ran the DNA. And what came back didn't match anything in the human reference genome the way it should have. And today, we're looking at one of the strangest stories to come out of ancient DNA research in years. A 2,000-year-old skull, a routine genetic test, and a result that sent researchers back to double-check their equipment because they assumed they'd made a mistake. This is part 1 of 3, so if you like stories where science stumbles into something nobody expected. So I'd hit subscribe now because we're going deep on this one. Elongated skulls are not new to archaeology. They show up across the ancient world, from Peru to Egypt to parts of Europe and Asia, and for over a century, the explanation has been well understood and thoroughly documented. Cranial deformation. Many ancient cultures intentionally reshape the skulls of infants, whose bones are still soft and pliable in the first years of life, using boards, tight wrappings, or cradle boards. It wasn't mutation, it wasn't disease, it was cultural practice, often tied to status, identity, or beauty standards, and it left behind skulls that looked genuinely alien to modern eyes, even though the people who had them were completely, unremarkably human. The skull at the center of this story fit that visual pattern almost perfectly. Long, narrow, with a sloping cranial vault that immediately drew comparisons online to the alien skulls that conspiracy communities have circulated for decades, particularly the famous Paracas skulls from coastal Peru, which have fueled ancient astronaut theories since the 1920s. It had sat in a collection for years, largely unstudied at the genetic level, treated as one more example of an already well-documented deformation practice. Curators knew what it looked like. Nobody had confirmed what it actually was, genetically, because for most of the 20th century that kind of testing simply wasn't available, and by the time it was, the skull wasn't considered a research priority. What changed was a research team decided to actually sequence its DNA, rather than rely on visual assessment alone, mostly as part of a broader project cataloging genetic diversity in ancient specimens pulled from museum archives across Europe. It was, by all accounts, meant to be a fairly routine addition to a much larger data set. Nobody expected it to become the centerpiece of anything. Ancient DNA work is slow, careful, and easy to get wrong. Genetic material from remains, this old, is degraded, fragmented, and extremely vulnerable to contamination from modern human DNA, which is present essentially everywhere researchers touch, breathe, or work. A single skin cell from a lab technician can compromise an entire sample if proper protocols aren't followed. Labs that specialize in this kind of analysis operate under strict cleanroom conditions, similar to the standards used in forensic crime labs, specifically to avoid a result that's actually just contamination from the person running the test, rather than a genuine finding from the ancient specimen itself. The team extracted material from the skull's petrous bone, the dense portion near the inner ear that tends to preserve DNA far better than other bone due to its density and protected location, and ran it through standard sequencing and alignment procedures. On it, comparing the extracted genome against the human reference genome used across virtually all modern ancient DNA research. In a normal case, ancient human samples align cleanly, if imperfectly, against that reference, allowing researchers to identify ancestry, health markers, and family relationships going back thousands of years. This sample didn't align the way researchers expected. A significant portion of the genetic material returned unusual or unmatched results when run against standard human reference markers, the kind of outcome that, on a first pass, looks less like this is a strange human and more like this doesn't cleanly fit our model. For a few uncomfortable days, according to people familiar with how these findings usually circulate internally before publication, the team reportedly reran the analysis more than once, assuming equipment error or sample mishandling, before anyone was willing to entertain what the raw numbers seemed to be suggesting. That's where we're going to pause for part 1. In part 2, we'll look at how this result got out into the world, why it spread so fast, and what other scientists said when they got their hands on the actual data. Stick around. Welcome back to part 2. If you haven't seen part 1, go watch that first, because we left off right as researchers were staring at a genetic result that didn't cleanly match anything in the standard human reference genome, and starting to wonder if they'd made a mistake somewhere in the process. It's easy to see why a headline like "Not Human" spreads fast once something like this leaks or gets reported prematurely. Ancient DNA studies rarely produce dramatic sound bites. They usually produce dense population genetics papers full of haplogroups and statistical confidence intervals that nobody outside academia reads closely. But an unusual sequencing result attached to an already strange-looking skull is exactly the kind of story that jumps from a technical preprint into viral territory within days. especially once it gets connected to decades of existing ancient alien folklore already built around elongated skulls from places like Paracas. Social media did what social media does. Screenshots of partial data, stripped of context, circulated with captions claiming scientists had confirmed a non-human species. Some posts leaned into full ancient astronaut territory, suggesting the skull belonged to a hybrid being or an entirely separate hominid line that had somehow survived undetected into the historical era. Others were more cautious, framing it as scientists baffled, which is often just as misleading because the researchers themselves were not baffled about basic biology. They were being appropriately careful about a messy result. What actually happened, according to the researchers who did the work and the wider community of geneticists who reviewed the data afterward, is far less exotic than not human, but arguably more interesting once you understand it. Severely degraded ancient DNA frequently produces mess, Y, partial, or ambiguous alignment results, especially when contamination, poor preservation, or low sample quality is involved. Unusual-looking results are common in this field precisely because the material is old, fragile, and easily corrupted, not because researchers are routinely stumbling onto non-human hominids in museum drawers across Europe. Independent geneticists who examined the released data pointed to a more mundane explanation: a combination of degraded sample quality and, in at least one widely discussed case involving elongated skulls, contamination with non-human genetic material introduced during earlier, less rigorous testing decades ago, long before modern clean-room ancient DNA standards existed. Older specimens have often passed through multiple hand-tests, multiple labs, and multiple eras of archaeological practice where contamination controls were minimal to non-existent. A skull excavated in the early 20th century, handled barehanded, stored without climate control, and tested with equipment that predates modern sequencing standards, is exactly the kind of sample where stray genetic material from animals, soil bacteria, or careless handling can end up mixed into the results. Once the sample was reprocessed under stricter modern protocols, the genome that emerged looked exactly like what you'd expect: fully human, W at the elongation explained by the same cultural head-shaping practice documented across dozens of populations worldwide, from the Huns of late antiquity to certain indigenous groups in North America to ancient communities in the Andes. There's also a pattern worth noticing here, one that shows up again and again in this exact genre of story. Nearly every viral non-human skull claim that has surfaced over the past 2 decades has followed the same arc. Wherever an early, poorly controlled test produces an anomalous result, that result gets reported or leaked before proper verification, it spreads across social media faster than any correction can follow, and then, months or years later, a rigorous reanalysis quietly confirms the mundane explanation to an audience, a fraction OF the size of the one that saw the original viral claim. By the time the real answer comes out, the not-human version has already been watched, shared, and believed by millions of people who never see the follow-up. That gap between how fast a sensational claim spreads and how slow a careful correction travels is arguably the most important part of this whole story. In part 3, we'll look at what the skull actually turned out to be once the dust settled, and why the real, unglamorous answer might actually be more worth knowing than the alien headline ever was. Welcome to part 3, the final part of this series. If you're just joining us, go back and watch parts 1 and 2 first. We've walked through how the skull was tested and how the results spiraled into a viral non-human claim before the actual science caught up with it. Now let's talk about what the skull really was. Peer-reviewed follow-up analysis on cases like this consistently lands in the same place. The skull belonged to a human being, genetically human, ancestrally traceable to known regional populations, with mitochondrial and autosomal markers falling within expected human variation once the sample was properly cleaned of contamination and reanalyzed under modern lab conditions. The elongation was cultural, not biological in origin, produced by intentional cranial bending in infancy, a practice researchers have confirmed through skeletal analysis, historical records, and even surviving depictions in art from cultures that practiced it deliberately for generations, sometimes as a marker of nobility, sometimes as a broader cultural or tribal identity. The not human framing that spread online traced back almost entirely to a mix of poor quality older testing, sensational reporting that outran the actual data, and the simple fact that partial or ambiguous genetic results, when described loosely or stripped of context, sound a lot more mysterious than they actually are. Scientists involved in re-examining these cases have been fairly blunt about it. An incomplete alignment, again. Saying a reference genome is not the same thing as evidence of a new species, and treating it that way skips over a lot of very ordinary, very well-understood biology in favor of a much more exciting story that performs better online. None of this makes the underlying archaeology boring, though. If anything, it's more remarkable than the alien headline ever was. Somewhere, the Yew Sands of years ago, a family wrapped an infant's skull in cloth or bound it between boards week after week because their culture believed a longer, sloped head signified something important. Maybe status, maybe beauty, maybe belonging to a particular group or bloodline. That child grew up, lived a full human life inside a body shaped by their community's beliefs, and was eventually buried in a way that preserved their skeleton for 2,000 years, only to end up misread by modern audiences as evidence of something otherworldly. It also says something important about how science actually moves, especially in an age where a single strange data point can outrun months of careful follow-up work before anyone corrects it. A strange result doesn't mean a breakthrough. It usually means more testing is needed, more controls, more scrutiny before anyone should draw conclusions. The researchers who reran the analysis under proper conditions did exactly what good science is supposed to do. They treated the surprising result with suspicion first, not excitement, and KPT testing until the explanation held up against scrutiny, even though the boring answer would never trend the way the exciting one did. The unglamorous answer, cultural cranial deformation plus degraded, possibly contaminated old D, nay, doesn't generate the same views as not human. But it's the one actually backed by the evidence, confirmed independently and consistent with everything else we know about how ancient cultures around the world shaped their children's skulls on purpose, for reasons that made complete sense within their own belief systems, even if they look strange to us now. So here's the real question worth sitting with. Now that you've seen all three parts, how many shocking scientists? Fic headlines you've scrolled past were built on a first messy result that got quietly walked back once the real analysis came in, long after the viral version had already done its damage. And how many of those corrections did you ever actually see? Let me know what you think in the comments. If you want more stories like this, where the wild headline meets a much stranger real explanation, the rest of the channel is full of them. Thanks for watching all three parts, and I'll see you in the next one.